For example, let us take this one. This sequence is cut at the G and A, guanine and adenosine. At that point, this restriction enzyme cuts this one and leaves some clip-like structures, or we can say cohesive ends or sticky ends. When this cut is made outside the center of the symmetry of the sequence, then it leaves the sticky ends. When the enzyme cuts both the strands at the same site, then it leaves bluff ends or blunt ends. In this way, some restriction endonucleases, they leave blunt ends. Some restriction endonucleases, they leave sticky ends. These restriction endonucleases, 4,000 have been characterized so far. Out of these 4,000 restriction nucleases, 600 are commercially available. The restriction endonucleases can leave blunt ends or sticky ends. For example, let us take this enzyme HIND3 or Haemophilus influenza D strain 3. It cuts this sequence AAGCTT, this sequence at AA. Always it reads the sequence 5 to 3 direction, and it cuts that sequence at A and A and leaves sticky ends. Here this sticky end has total how many? 1, 2, 3, 4 nucleotides are there here. Here also same, but in a reverse direction. They are looking like clips. So we can call them as clip ends or cos ends or cohesive ends. In the same way, let us take one more enzyme, PST1. This PST1 cuts the DNA molecule in between G and A, and it also leaves protruding ends. So thus, the sticky ends or cohesive ends are produced. And one more enzyme let us take, which is obtained from Escherichia coli, R strain 5, ECO R5. It cuts the DNA strands, both the strands at the same point, and it leaves blunt ends. In this way, the restriction endonucleases can cut the DNA sequence and form cohesive ends or sticky ends, or they may leave the blunt ends. That depends upon the restriction endonuclease that we use on DNA, a specific DNA sequence. What are the properties of restriction enzymes? These cut DNA into defined fragments, and these are generally called chemical knives or molecular scissors. The first restriction endonuclease used in genetic engineering is HIND2, Haemophilus influenza D strain 2. This Haemophilus influenza D strain 2 cuts a specific sequence 5 to 3, 5 prime to 3 prime, and GAATTC. Another strand is like this, the complementary strand. At the always it cuts this sequence in between 5G and A. So always it cuts it 5 to 3 direction, and between G and A it cuts. Suppose look this one. Here AG is there, and it will cut here 5 to 3. It reads in this way, it will cut here, and it forms sticky ends HIND2. And these restriction endonucleases cut palindrome sequences of about 4 to 8 nucleotides length. Palindrome sequences are the sequences that we can read from both directions. For example, let us take this is a palindromic sequence GAATTC. This is a 5 to 3 direction, and look this one. The opposite one is the same GAATTC. So this is the palindromic sequence. If we read it from both the directions, it gives the same meaning, or it has the same wording. Those are called palindromic sequences. One more example is the Malayalam, and that is also a palindromic word. These are the palindromic sequences, but the palindromic sequences contain 4 to 8 nucleotides length. According to the books, 900 restriction enzymes are extracted from 230 prokaryotic strains. It means one prokaryote may have more than one restriction endonucleases. These are some properties of restriction enzymes. One is it cuts the DNA into defined fragments, and it cuts specific sequences, especially the palindromic sequences. The length of the palindromic sequences may be range from 4 to 8. Always restriction endonuclease reads the sequence from 5 to 3 prime direction. These are described as chemical knives or molecular scissors. The first restriction enzyme used in genetic engineering is HIND2. So far, 900 restriction enzymes are extracted from 230 prokaryotic strains. The first letter stands for genus of that organism. Here, this is obtained from Escherichia. So, the genus Escherichia, in that one first letter is E, that letter we have taken here. The second and third letters stands for species. The species is coli. In this one, CO we have to take, and 2 letters from the species we have to take, and thus it becomes ECO, Escherichia coli. R is the strain, and this last number, Roman number, order of that enzyme extracted from that organism. Let us take this one. In this case, 1 is there. This 1 indicates that this was the first enzyme obtained from E. coli. Thus, the first letter indicates the genus, the second and third letters represent the species, and the fourth letter shows its strain. The Roman number indicates the order of enzymes extracted from that strain of bacteria. And one more example, let us see. Here, HIND2 is there. H stands for Haemophilus, IN stands for influenza, and D for strain, and 2 is the second enzyme extracted from this Haemophilus influenza D strain. So in this way, the naming of the restriction enzymes is carried out. Some restriction enzymes mentioned for understanding, and in that one, let us take BOM H1. B for Bacillus, AM for amylo liquefaciens. H is the strain, and 1 is the order of that enzyme extracted from that strain. And it cuts the this sequence GGA TCC sequence at 5GG and leaves the sticky ends. The same way, let us take ECO R1. E for Escherichia, C for coli, R is the strain, and 1 is the first enzyme obtained from that bacterium. It cuts in between G and A. The sequence that it can identify is GAATTC. And in the same way, let us take HIND3. So far we discussed some enzymes. In that one of them is the HIND2. Here this is the HIND3. Haemophilus influenzae. H for Haemophilus, IN for influenzae, D is the strain. This is the third enzyme obtained from it. It cuts in between A and A, and it can recognize this sequence. Let us take HIND2. This HIND2 is cutting between G and A. It can recognize GAA TTC. But HIND3 can recognize AAG CTT sequence. That is GAA TTC. This is the AAG CTT sequence it can recognize. Next one, PST1. P for Providencia, ST for Stuarti, and 1 is the enzyme obtained from that one. It can read this sequence, palindromic sequence, CTG, CAG, and it will cut in between A and G. Sol1. S for Streptomyces, AL for Albus. This is the first enzyme. It can read GTC, GAC, and at this junction it can make a cut. Next one, Serratia marcescens. S for Serratia, MA for marcescens species, and 1 is the enzyme. It can recognize this sequence CCC, GGG. And look here, these enzymes are leaving sticky ends whereas Serratia marcescens leaving blunt ends. Caryophynon latum, CLJ1. This one is leaving sticky ends. And Proteus vulgaris, PVU1, the enzyme obtained from that species. It can cut between the C and T. These are some restriction endonucleases which are given in our book. These we can see in at the cloning sites of the PBRC22 and PUC plasmid. In this, the very important restriction endonucleases are these three. BAMH1 for examination point of view. These are very important. Bacillus amyloliquefaciens and its sequence, and EcoR1, Escherichia coli, HIND3, Haemophilus influenzae. These are very important, and all these are leaving the sticky ends. And one more is there, no, HIND2, and which acts on GAA TTC. That is the first enzyme which was used in the genetic engineering. That is about the restriction endonucleases.